morning. I'm Dipan Shah, and welcome to Methodist DeBakey Heart and Vascular Center Grand Rounds. Today, I'm very pleased to have with us Dr. Miguel Valderrábano. Miguel is the chief of the Division of Electrophysiology here at Houston Methodist DeBakey Heart and Vascular Center. Miguel completed his medical schooling in Spain and then came to the United States to the West Coast, where he did his internal medicine residency at UCLA and his cardiology and EP training as well as research training at Cedars-Sinai. Miguel has really been a pioneer in use of alcohol for electrophysiology procedures, and he's the national PI on the recently completed vein of Marshall study for atrial fib ablation. So we're very pleased to have Miguel with us today, and he's going to actually give us a little bit of kind of the journey that he went on, and really the concept of taking an idea from being in your mind, maybe when you're taking a shower or something, and to actually putting it into a randomized clinical trial. And so Miguel, we're very pleased to have you with us today, and we look forward to hearing about your journey.
Thank you. So when Dr. Zogmi asked me to share my trials and tribulations running this trial, I thought it was a great idea, something that I had actually had in my mind to discuss, because it's been quite a learning experience for me. And the whole thing starts with the clinical limitations of the procedure we do. We make a living out of doing AFib ablations, and AFib ablations are based on this mechanistic paradigm that it's ectopic beats from the pulmonary veins that trigger atrial fibrillation, and if you create lesions to isolate electrically the pulmonary veins, you would achieve cure. This was pioneering work by Hasager and others more than 20 years ago, and it's been the basis of AFib ablation. And the outcomes have supported this use, at least for paroxysmal atrial fibrillation. There's no doubt that atrial fibrillation ablation with pulmonary vein isolation achieves better results than any drug. And it doesn't really matter whether you use radiofrequency or a contact force sensing catheter or cryoablation or laser ablation. The bottom line is that for paroxysmal AFib, pulmonary vein isolation works. The problem is when you look at persistent atrial fibrillation, the outcomes are quite unsatisfactory. And depending on the series you look at, the recurrence rate after a single procedure could be as high as 71%. So that prompted us to look into other ways to tackle persistent atrial fibrillation. And, you know, we should think as physiologists, and our approach was, well, what else can we tackle that could be playing a role in persistent atrial fibrillation beyond pulmonary vein ectopy? And that got us thinking into the autonomic cardiac nerves. And the left atrium is coated with several plexuses of nerves that modulate atrial physiology. And they can make the atrial tissue more prone to fibrillation by shortening the action potential duration and other mechanisms that I won't get into. But interestingly, in experimental models of atrial fibrillation, discharges from these nerve structures were shown to precede atrial fibrillation. Oh, let's see if we come back. Okay. And keep in mind, these nerve plexus are located in different areas that are relatively consistent anatomically around the ostia of the pulmonary veins and in a nice and distinct structure called the ligament of Marshall. This is the remnant of the left superior vena cava that during embryogenesis undergo atresia, and it's a twig that connects with the coronary sinus and is full of innervation. In animal models of atrial fibrillation, nerve discharges from the ligament of Marshall were shown to precede the onset of atrial fibrillation. And that was the background, the mechanistic background that was the basis of this idea. And I hope my computer behaves. Anyway, so a little bit more details about the vein of Marshall anatomy and histology. Vein of Marshall is in the epicardial aspect of the left atrial ridge, a structure that all echocardiographists are very familiar with because it, I don't know what's going on there, okay. It's the coumarin ridge that you see on transesophageal echocardiogram. And it's between the left-sided pulmonary veins and the left atrial appendage. And As you can see it here in this autopsy specimen, it's a distinct vein on the epicardial aspect of the left atrium. And on histology, is full of cardiac innervation, both adrenergic as well as cholinergic. These are stains for tyrosine hydroxylase and acetylcholinesterase, which stain for sympathetic and parasympathetic innervation, respectively. And the vein of Marshall is surrounded by bundles of nerves. And even in clinical cases, the vein of Marshall had been shown to be a trigger for atrial fibrillation. So in patients with paroxysmal AFib, you could see that in some cases, the first beat of AFib would arise from the vein of Marshall. So this was background knowledge that my mentor at Cedars-Sinai was very interested in and had done a whole line of research. And I did not really play much part in that research, it was just background knowledge. But when I moved to Houston, I had a conversation with Dirar Khoury, who was an engineer working here in the Department of Cardiology. And he was interested in imaging of the left atrium. And he asked me, how can I inject ultrasound contrast in the left atrium? And I started thinking, well, listen, the atrial arteries are very inconsistent. They're small, and I don't think you're going to have a consistent atrial artery that you could inject echo contrast. But the vein of Marshall is always there. And then in the same conversation, I thought, wow, well, we could put a catheter in the vein of Marshall, we could inject echo contrast, and why not alcohol? So that's how the idea came to my mind. And then I remembered meeting with my mentor when I was a first-year fellow, and I would always keep asking him questions and showing him ideas. This is Peng Chen, the current editor-in-chief of Heart Rhythm Journal. He said, after being fed up with me, he said, listen, Miguel, the difficult part of research is not having ideas. We all have ideas. The difficult part of research is the paperwork. So if you stick to the paperwork, if you're able to take the idea and present it and get funding and defend it and write it up and write a protocol, all the paperwork that follows an idea is what will make it succeed or not. So with this idea, the first thing we did is we needed to get money. And we got the money first from an unusual source, the Houston, Texas football team. Apparently, Methodist had a deal with the Houston Texans whereby they would make a cheerleader's swimsuit calendar and sell the calendar, and the profits of selling calendar of half-naked women would come to Methodist for research support. And we had a whole ceremony in the stadium where they gave me this check, and you can see this is Alan Lumsden here and Mike Quiñones here and the cheerleader here. And they gave me this check to fund this research. And what we did first was animal studies to see if this could be done. And of course, it was critical to pick the right animal model. Pigs don't have a vein of Marshall, canines do. So anyway, we tried in 17 cases, and in 10, we could find a vein of Marshall, and we could put a wire. You can see a coronary sinus venogram. The vein of Marshall is this little twig. We could put in a wire in that little twig and then put a balloon. And we could deliver alcohol and get the procedure done. First time it took us 8 hours, and Daryl Schultz here in the animal cath lab was instrumental in getting this done. And sure enough, when we get in there and we put a balloon and inject alcohol, we create a scar. And these are maps of the left atrium with the pulmonary veins are these tubes. And after alcohol, we would create an area of low voltage shown by the red area here. And interestingly, we could achieve some local regional denervation. So what we did is we exposed the vagus nerve and stimulated the vagus nerve. And we could show, after this stuttering here, we could show, as is expected, when you stimulate the vagus nerve, you get a shortening of the refractory period. That's normal physiology. And that's mediated by vagal connections from the vagus nerve into the left atrium. Where after alcohol, we blunted that response, consistent with some form of local regional denervation, which was one of the things we expected with this procedure, that we would eliminate not just myocardial activity and create a myocardial scar, but also eliminate the local innervation that triggers AFib. And when we look at the specimen macroscopically, we could see this is the vein of Marshall going up and the ligament of Marshall upstream. This is the left atrial appendage. You could see this area of pale tissue after alcohol delivery, and you can see on the endocardial aspect as well. So we got excited, and then I said, well, let's just get this in humans. And I wrote an IRB protocol. The first time I was writing an IRB protocol, and it didn't last long. I was tabled immediately. It was returned to me with a rejection. Basically, they say, well, there's not enough explanations. Why are you doing this? Where is the vein of Marshall? What does it mean? How are you going to make sure this is safe? What are you talking about? Possible CS dissection? What's a dissection? What's a CS? So CS meaning the coronary sinus. So, okay, went back and I rewrote the protocol. And then I asked for a 10-patient protocol, just a phase 1, first-in-man protocol. And they said, okay, do one. Let us know before you do anything. And contact Susan Miller, Al Reisner, and Greg Pratt before doing the first procedure. And immediately after you finish, let us know how that person does. And if the person does okay, we will approve one by one up to the 10 you requested. I thought, okay, I'll take it. So we did the first patient in May 7, 2008, and sure enough, the patient had a nice vein of Marshall. As you can see here, we could cannulate it and put a balloon and injected alcohol. The truth is that the first time I injected alcohol, it was scary because I wanted to see, going back to the original idea of the Arcuri of injecting echocardiographic contrast in the vein of Marshall, and the first thing I saw was on intracardiac echo, the left atrial cavity got full with contrast. And I thought, gee, I'm going to give a bolus of alcohol directly into the left atrium. That's going to go to the coronaries. The patient's going to die. I'm just going to die myself. So I paused for a minute and really gave the alcohol slowly. The idea is that the alcohol has to go through the myocardium before going into the left atrial cavity to create this tissue ablation. And some veins are huge with a lot of capillaries, like this example. And some others are smaller without capillary network. So yes, you can get a rapid injection of alcohol into the left atrial cavity. And the point is here, just inject it slowly over 2 minutes and you'll be fine. We did 2 cases. We had the animal study, all the animal data prepared, and we published it in Circulation EP in 2009. Then I said, okay, it's time to get more serious funding, and let me apply for NIH funding. And we did. And of course, the first setback came when the grant, which was a small grant, a 2-year grant, was rejected. And it was percentile 54, as you can see here. And when was the last time you were in the bottom half of anything? It's kind of bad and disheartening. But the main concerns the reviewers had were that they were concerned about safety. And okay, if safety is the issue, then we can create mechanisms to appease the concerns of the reviewers and guarantee patient safety. In the meantime, we did publish 2 papers, one to show the initial feasibility and a second one to show the adjunctive ablation effects by alcohol. And we wrote a protocol for a randomized trial and sent it to the IRB, still without funding, but the idea is to get the ball rolling. And when we submitted to the IRB, we got the second setback. Now, by this time, we had already been enrolling patients in this phase 1 study. We had completed a 10-patient study. And this took us about 8 months or so. At that point, the Research Institute hired a new regulatory person. And when that regulatory person looked at my submission of a randomized study, they said, alcohol, that sounds like a drug. Is this approved for this indication? Maybe not. And they say, maybe you need an investigational new drug application approved by the FDA. And I said, geez, alcohol has been used since the 1980s. What are you talking about, a new drug? And they said, well, the FDA may be concerned that you may have commercial interest in alcohol getting a new indication. I was just completely stunned. But anyway, the bottom line is that we asked the FDA, and they said, yes, you need to apply for an IND. And IND submission to the FDA is not a minor thing. They want a lot of documentation. And when they asked for documentation, I said, hey, look at these 2 papers, they're published. They said, no, we want a folder for each patient you've done, the H&P, the procedure note, the follow-up notes for each of the patients you have done, and a bunch of paperwork. So I said, okay, I'll take care of that. The funny thing is, when the FDA said, yes, you need an IND for this, the IRB, which had approved this study for now almost 9 months, they said, no, you can't, we reverse our approval. This protocol is not safe. So what are you talking about, not safe? It was safe last week. Anyway, the bottom line is that we had a halt for a long time until I got the paperwork for the FDA, IND submission. The IND was approved, and then the IRB approved again for a second time the same protocol. The nice thing about these delays was that by the time I resubmitted to the NIH an application for this R21, this small grant, I had an IND, I had 2 papers, and I had all the paperwork that would satisfy the concerns of reviewers in terms of safety. And it went from percentile 54 to percentile 1, meaning it was the top grant in that study section meeting, which is nice. We got the money, and we continued doing more kind of mechanistic work in patients. So we did work looking at whether the vein of Marshall electrical activity gets ablated when you do an extensive endocardial ablation with conventional catheters. And we looked at 54 patients, and sure enough, all 54 had preserved vein of Marshall electrical activity, even despite previous aggressive ablations. And that electrical activity could be eliminated with alcohol. We also described connections between the vein of Marshall and the pulmonary veins that could be bypassing regular endocardial ablation. And I won't get into the details of these tracings, because they're basically for EPs. But the idea here is you see in this RAO view and this LAO view, the coronary sinus is here. This little microelectrode catheter is in the vein of Marshall, and we could show that there were direct connections between the vein of Marshall and the pulmonary vein where we have this circular catheter. And also interesting, when we injected alcohol in the vein of Marshall, we could achieve electrical disconnection of both the superior and the inferior pulmonary vein, depending on the cases. And in some cases, indeed, we could show that there was electrical firing from the vein of Marshall, triggering runs of atrial tachycardia in patients that had failed a previous ablation, and that after alcohol, we would terminate that and get rid of it. We also showed that we created a scar from the mitral annulus to the coumarin ridge. And this is an MRI done here. I think, Dipan, you must have read this one. Pre-alcohol, you can see there's no enhancement. And after alcohol, this bright area in the left atrial wall going from the pulmonary vein, the coumarin ridge, and all the way to the mitral annulus. And this you could see on intracardiac echo as it gets echogenic. The coumarin ridge gets echogenic here. And you create a scar that goes from the mitral annulus to the left inferior pulmonary vein. Now, that is important because a common mechanism of failure of ablation is the patients come back with perimitral flutter. That is electricity spinning around the mitral annulus. And the way we treat it is by creating a line of burns from the pulmonary veins to the mitral annulus to prevent that circuit. And that is technically difficult because the coumarin ridge is there and is very thick. Well, we showed that with alcohol in there, we could create a nice lesion very quickly. And remember, it's epicardial to endocardial because the vein of Marshall is on the epicardial side. And we could achieve a nice block in that area that would terminate the flutter. We created a scar. We quantified how long it took to get the procedure done, how much fluoro time it took, and all these things that kind of set the stage to establish feasibility of the technique. These are examples of perimitral flutter. I won't get into the details, but the idea is that for every perimitral flutter that we found, we could demonstrate that the vein of Marshall was part of the circuit and actually eliminate that. And we had a series of publications about this, setting the stage for establishing a clinical utility of the vein of Marshall alcohol, particularly for perimitral flutter. Remember, as I was saying, the ablation of the mitral isthmus, sometimes it's tough with regular radio frequency to create bidirectional conduction block. And in a series of 77 patients, we achieved a block in all of them only requiring 2 minutes of radiofrequency to get this done. So with this kind of work that we do while we are trying to get more money for a grant, I wrote a grant application for a randomized clinical trial. I got some collaborators to agree to be part of the trial. Mostly here at the VA and Ben Taub agreed to be part of it. And we sent an application, and it was rejected. I got a priority score of 45. And again, the main issues they had here was not just safety concerns, which they did have safety concerns, but they also had concerns about the clinical trial administration. And I had no experience in that. I had been part of clinical trials, but I had never run a clinical trial. And you have to convince the NIH that you have the structure for running a clinical trial. And it's not just writing the right kind of gibberish, which I did. I said, well, yeah, the Research Institute at Methodist has all the infrastructure required for a clinical trial. Sounds good, but that's not enough. You need to tell them that you have a dedicated room where you're going to have the patient folders, and that room is locked at night, and that you have secure server where you're going to store the data, and you want to show them how you're going to collect the data. You don't just say, yeah, we will collect the data. No, they want to show the forms. They want to see the forms that you're going to collect the data with. They want to see all this paperwork, really, that has to be convincing enough so that they know that you're not going to waste their money. And indeed, I thought this is not really scientifically interesting to describe how you're going to collect paperwork, but it is what makes or breaks a grant when it comes to a clinical trial. So anyway, the grant was rejected. I kept doing some work, and we did some interesting work. I think probably our highest impact paper was in JACC 6 years ago, where we showed that we could actually get in the vein of Marshall and stimulate the cardiac innervation in humans, and show that by stimulating the nerves around the vein of Marshall, we could create a systole. So this was fascinating because the vein of Marshall is all the way lateral in the left atrium. The AV node is in the opposite side of the left atrium. By stimulating the vein of Marshall, we could actually arrest AV node conduction through interneurons connecting the nerves of the vein of Marshall with the nerves that control the AV node. And in cases like this, you have 6 seconds of a systole with stimulating the vein of Marshall. And when we gave alcohol, we tried to do the same thing, and we could not show it. So we showed that we had denervation. So we get another paper, and we say, okay, I need help to get this grant done. And I asked around, and in a hallway conversation with Christy Valentine, he was kind enough to sit with me for 5 hours and go line by line over the criticisms of the NIH and help me get a new application. And when this was sent, it was finally funded. So it was finally funded, and then I get the money, and it's great. Now I need to come up with a name for the trials. And we had 2 trials, one for de novo ablation, where patients will get randomized to vein of Marshall alcohol combined with ablation versus just ablation. And then a second trial for recurrent, so previously failed ablation. So Venus and Mars were the terms that we came up with. And this was not intentional, but when I Googled if there was any other trials for Venus and Mars, I found a bunch of classic paintings from the Renaissance, some of them even sexually explicit, like this one where Venus and Mars are at it there, surprised by the gods. This was completely unintentional. It was kind of interesting. But anyway, Venus and Mars was a catchy term that stuck. And we wanted to randomize patients to either pulmonary vein antral isolation, the standard approach, alone or combined with vein of Marshall alcohol. And I'll just stick to Venus, which is the one we have completed. The primary endpoint was freedom from AFib or atrial tachycardia greater than 30 seconds duration after a single procedure, without the use of drugs, and after a 3-month blanking period. This is kind of the standard in clinical trials of AFib. We had some secondary endpoints, freedom from AFib after more than 1 procedure or on drugs. The AFib burden is an important parameter, how much time the patient spends in AFib. Remember, we started with persistent AFib, so patients that had AFib of at least 1 week duration. And we had some procedural parameters to measure the need for repeat procedures and recurrence as atrial fibrillation versus atrial tachycardia. We included patients with age between 18 and 85 with symptomatic persistent atrial fibrillation, as I described, and refractory to at least one antiarrhythmic drug. We excluded patients with previous ablation attempts in venous. In Mars, you have to have been a previous ablation failure. And patients with extreme left atrial enlargement and with decreased ejection fraction. There was an EKG core lab that was blinded to the randomization outcome. And the patients were also blinded. Obviously, the operators were not. And we would randomize in a 1 to 1.15 ratio. The idea here was we expected a 15% rate of technical failures of the Weill-Marshall procedure. That is, that we could not get in the Weill-Marshall or we could not complete the ethanol infusion. And the idea is that we would have enough patients treated with alcohol so that we would have a kind of a 1-to-1 as treated comparison. Patients would be followed 1 month, 3 months, 6, 9, and 12. And at 6 months and at 12 months, we would do a continuous 1 month monitor. This is more intense monitoring than other clinical trials that have been published to date, where they just do 48-hour holters. So this was 1 month continuous monitoring. Of course, if patients had implantable monitors, that would replace the continuous monitoring, and that would be even better. And patients would be considered failures if they had either clinical recurrences of symptomatic atrial fibrillation or tachycardia, or they had more than 30 seconds of atrial tachycardia or fibrillation in the monitor. So very, very strict. And with these numbers, 156 in the catheter ablation group and 180 in the Weill-Marshall group, we achieved 91% power. So we get this going and we start recruitment, and this is when more setbacks come into my life. So we planned to enroll in venous 336 patients over 4 years. That was this green shaded area. In blue is the actual enrollment. Of course, when you plan a kind of a linear constant enrollment, but that doesn't happen in any clinical trial. And you can see this red line is the percentage of expected enrollment over time. At some point, 1 year well into the trial, we were as low as 23% of expected. So we're really, really low. And at that point, I requested the IRB to let me open new sites. At that point, we were getting some papers out. There was some interest in the EP community to be part of this. So I had some friends that wanted to be part of it. I said, fine, just send me your stuff. Let's see if we can add you as a site. The NIH was okay with that. But when I requested the IRB to open new sites, I said, no. No, and in fact, we want you to close Austin, one of the enrolling sites. I said, why do you want to close Austin? Well, because they failed to get into the Weill-Marshall. But I said, yeah, but that's okay. That's what we planned that. If we fail, that's fine. I want to know that. If it's not a failure of the trial, if the operator cannot get in the Weill-Marshall, if the conclusion of the trial is that this is not doable, it's still a valid conclusion. Don't close the site. Anyway, after going back and forth with the MRI, we finally get new sites added, but they don't perform. And then we get the monitor reports, and the monitor reports come with too many protocol deviations. So they say, stop enrolling. You need to fix this. And I say, I can't stop enrolling. I'm at 25% of expected. Like I should have, at this point, I should have 100 and some patients, and I only have, 40. This is a mess. So in the meantime, the PI, so myself, gets reported to the NIH by this institution as a non-compliant investigator. So I'm getting hit everywhere. The FDA, and so the NIH wants me to enroll more patients. The IRB doesn't want me to add more sites. The institution reports me as being non-compliant to the FDA and to the NIH. So this was miserable times in my life. And because I don't enroll enough, the NIH says, listen, unless you get your act together, we're going to take the money away. So this was a pretty miserable time. Let me just show you the protocol deviations that prompted this accusation of non-compliance. most of them were, of course, irrelevant. The final outcome of this accusation was a dismissal by the FDA. But the wording that got to the FDA was that Dr. Valderrábano has demonstrated multiple instances of non-compliance in his conduct of the study, and we have made a final determination of continuing non-compliance. It's pretty disheartening. And these were window deviations. So the patient will show up at 2 weeks or whatever, 2 weeks later after his 1 month appointment, and that's a protocol deviation. It was my mistake because I didn't have windows incorporated in the protocol because I was basically completely ignorant of how to run a clinical trial. Missing data was a problem. Okay, so I'll try to fix the missing data. A patient doesn't show up to their 3-month appointment and I don't have the EKG, but I know that the patient went to his doctor. So I said, well, let me get the EKG from the doctor's office. I put it in the file, and because the EKG from the doctor's office says, medical office of New Mexico somewhere, that's not an approved site to conduct research. So that's another violation. And I said, what do you want, missing data or a violation? Just give me a break. It was constant. Every week I would have fights with the IRB. The most, I think, flagrant was this what they said was an exclusion criteria violation, because a patient was participating in other studies and that was an exclusion for a clinical for entering the clinical trial. And then I said, what clinical study is the patient part of? And then I read the chart and I read that the patient had undergone a sleep study to diagnose sleep apnea. And I said, that's a diagnostic test. It's not a research study. So this kind of pedestrian things I had to deal with. Missing initials in my documentation. Another ridiculous one was not documenting the ethanol storage temperature. And I said, well, I didn't know do we have to refrigerate it? No, the package insert says store at room temperature. Do I need to document the temperature or that is in a room? Like if I store it in a hallway and not in a room or in the stairs, is that a protocol violation? It was crazy. It was crazy. My conclusion out of this was the IRB was not interested in details. The paperwork completeness is paramount to them. And the research administration of the hospital is there to protect the research line of business of the institution, and clearly not the investigator. And when the list of protocol deviations is too long, nobody cares about reading the list. You're not compliant. So this was a pretty miserable time in my life, but thankfully, my colleagues and co-investigators came to the rescue and started slowly enrolling more and more patients, and we started getting better numbers. And very important was the role of Raquel Bunge that was instrumental. Basically, she came to the project and she became my liaison with the IRB. Within a month, we were all friends and everybody was happy and the paperwork was taken care of. And we could continue the project in a relatively harmonious way. So the trial conduct is shown here. We completed it. We enrolled 158 patients in the catheter ablation only arm. Some were missing, 142 in the primary analysis. In the Veno-Marshall group, we enrolled 185, and we failed Veno-Marshall ethanol in 30. It's a little bit more than the 15% that we expected. And we concluded we had patients for the intention to treat analysis as well as the per-treatment analysis. So great, we have all the study done. Let's look at the data. Well, setback number, whatever. As we prepare for the statistical analysis, the research administration decides that they need to remodel the statistics groups, and my statistician is fired. So I have all this data. I have to prepare a report to the DSMB. I'm very excited to write the final results, and I don't have a statistician. So at that point, I think back of the picture from the 1600s, and I see that I feel like Venus and Mars trying to get business done here. And then the research administration, the FDA, the IRB, everybody's looking at us and trying to interfere. And there's some help from cardiology, help from the NIH, but it really felt like that these people were not really helping. Anyway, with some help, which I will explain later, I got a lot of help to get this done. We finally completed the statistical analysis and reported this in the ACC meeting. And the primary endpoint, which was ablation success, freedom from AFib or atrial tachycardia more than 30 seconds after 3 months, single procedure of antiarrhythmics. In the catheter ablation group, was 38%, and in the intention to treat veno-marshal was 49%, and in the per treatment, so the successfully treated veno-marshal patients, 51%. So this gave us an odds ratio of intention to treat 0.63, p-value 0.037, and in the as treated group, odds ratio of 0.57 with a p-value of 0.05. So the recurrence was, decreased by 43% when you got the alcohol on. So happily, the procedures seem to have an impact on the outcomes of AFib ablation, which is what we were going after. If you look at the components of the primary endpoint, if you just look at recurrence of atrial fibrillation and tachycardia, excluding deaths and missing data, 52% of the patients in the catheter ablation group recurred versus 41 in the intention to treat veno-marshal group and 40 in the successfully treated veno-marshal group. So that was really nice. We had an impact in the clinical success of ablation. And if you look at the time to recurrence, the Kaplan-Meier curves were still significant. As treated, I show it here, a hazard ratio of 0.66 with a significant p-value. So it's nice. It's not a miracle. Obviously, by no means we have a magic bullet for AFib, but clearly it makes an impact. Other secondary endpoints were important after repeat procedures. The success was 65% as treated versus 53 in the conventional catheter ablation, and atrial fibrillation burden became a little bit tricky to analyze statistically. I'll show you a figure in a minute, but it was better. It was more reduced. The percentage of patients with zero AFib burden was higher in the veno-marshal treated group. And the need for repeat procedures was decreased. So 17% in the veno-marshal group had to get a second procedure versus 25% in the conventional ablation group. This is the atrial fibrillation burden divided in three groups: zero burden, 0 to 5% versus more than 5% atrial fibrillation burden. The percentage of patients that had zero burden was 78 in the catheter ablation group versus 68 in the conventional catheter ablation. Adverse events were similar. We had perhaps more incidence of fluid retention after veno-marshal alcohol. I can't really tell you why that is. Maybe some increased stiffness of the left atrium that make the patient retain fluid after. The bottom line is that we can conclude that in persistent atrial fibrillation, veno-marshal ethanol added to catheter ablation reduces the recurrence of atrial fibrillation and tachycardia, reduces the AFib burden, and may reduce the need for repeat procedures. There's obviously some limitations. The veno-marshal procedure was only completed in 83.8% of the patients, and it may increase the risk of fluid overload. So this was the final presentation we did at ACC. I kind of closed this project. Again, the paper is written and is accepted for publication. It will come out in late October. But thinking back, when you have an idea and you want to make a contribution, I think the key part here that determines whether you're going to succeed or not is motivation. And you have to have the right kind of motivation to get this through. Sure, having an idea requires some smarts, but not a lot. I mean, you just need to be curious about what you're studying. You have to have a passion for what you're doing. Otherwise, you don't really care about the gaps of knowledge. You need to obviously know enough about your field to have meaningful ideas, and you have to integrate the information. You have to be connecting dots all the time. Some inspiration is required, but inspiration comes, kind of randomly. But once you have an idea, then to take the idea to fruition, the key, I think, is resilience. And resilience comes from motivation. And the motivation in some of us is because you want to make a contribution to the field. Do you have scientific ambitions for the sake of science, or just want to write a paper and show it to your mother and brag about it? Some people have entrepreneurial ambitions, and those are fine. Some people just want to patent, start a company, and change the field that way, and those are legitimate motivations. But the key here is some motivations get you this far, some other motivations will get you farther. Some people are motivated just to be prominent in the field and make money as key opinion leaders and to be in the podium. or to attain leadership positions. Those are fine, but you gotta be honest with yourself as to what it is that is driving you so that you can expect how far you're gonna go with an idea. Some people just wanna do research to get into a fellowship program and then go on to private practice. That's fine, that'll get you only that far. The key here is be honest with yourself and know what it is that is driving you, because then you can expect how much effort you're willing to put. Because from the idea to the accomplishment, it's just about resilience and faith in what you're doing. Because nobody will fight for your idea more than yourself. It is key when you get in trouble or when you're in difficulties to seek mentorships and allies. And I have a few names that I have to put out here because they were instrumental. So Christy Valentine was key in us getting the grant. That afternoon that we spent together reviewing the criticisms was key. And then Dr. Q, Dr. Lamson, and Dr. Phillips were key to help me out when I was fighting with everybody. IRB, FDA, you name it, everybody was on my case, and I was punching back inappropriately sometimes. And then Dr. Zobi was helpful getting me statistical support at the end when things were about to collapse, because after 10 years of work, I had no statistician. And it's very important that you engage like-minded collaborators. You cannot get these projects of this size alone unless you share the passion with other investigators that are willing to put work with you. Do not dismiss the paperwork. This is what I did, and it was a big mistake, because paperwork is the passion of those without ideas. And just like you have passion for your project, they have passion for it, and you have to respect that. And do not dismiss administrators and regulators. You want them on your side. And most of them actually can turn out to be, at the end, effective partners with you. And even though your project is personal to you, you should avoid engaging in personal battles. That's what I have. And then this is a... Pick yourself up, dust yourself off, start all over again. This is...
No one could teach you to dance in a million years.
This is a good conclusion. Just pick yourself up, dust yourself off, and start all over again. Thank you.
All right. Wonderful. Miguel, that was a fantastic overview. And first off, we've got room in the audience. I mean, we've got opportunities for questions from the audience. So please text your questions to 37607 and text DeBakey to get the text started. Or you can go to pollev.com/DeBakey. So we look forward to some questions from folks that are listening. I'm going to start off with a few questions. So first off, I was a little bit worried with today's Grand Rounds that you were going to be very mellow and not give us the full truth. But as we know, Miguel, you're not afraid to put it all out there, which we appreciate. And I think it really shows 10, 12 years of your life and the trials and tribulations that you've had to gone through. And to me, it seems like it's all started with alcohol and Texans cheerleaders is where this whole thing got started in the end. Let me ask you this, a couple of questions. One is, what are the next steps? I mean, obviously, you've done this. I think this is really kind of pioneering, groundbreaking work. What are going to be the next steps?
So there's a few next steps. And this is what I think the most fascinating part of any research, that even when it could have failed, and it didn't fail, but that's, to me, is anecdotal. In the process, we've learned so much. So in 2011, I had a patient with ventricular tachycardia that we could not get. 3 ablations, we ablated epi, endocardial, we were going nowhere. It was deep in the septum. I thought, gee, I get into the vena marshal all the time. Instead of going to the left, let me go to the right in RAO. And then I got into a ventricular vein and got signals from deep in the muscle of a ventricular vein and gave alcohol and took care of it. So all of a sudden, this is just connecting the dots again. I say, okay, if I do this in the atrial vein, can I do it in a ventricular vein? Now we have, we just published a series of 63 patients that we treated, 55 of them successfully. We treated VT by using alcohol in ventricular veins. So that's one spin-off of the procedure that it's leading to a whole new project. And another spin-off is the ability to get signals from the vein of Marshall. It's not just, doesn't just allow us to get atrial signals, but because it's full of nerves, we can actually start recording nerve signals. And that has not been done in vivo, but mechanistically it opens a whole new arena of understanding arrhythmias if you can actually discern the role of innervation triggering arrhythmias. So that's one other project. So it's fantastic because if you're engaged in what you do, there's always opportunities. You know, even if the veno-martial outcomes, alcohol would have been a failure, even if the venous trial would have been a failure, I still had this ventricular side or the nerve side. So failures are never complete failures. And you know, sure, we have success and it probably is useful and I use it, the technique. But what matters now is that I have now work to do in the next 10 years for something else that came out of this.
Yeah. And so, you know, you told us about the first procedure you did, which I think you said was about 8 hours or so. Tell us about the learning curve and what's the time to do the procedure? What's it down to now?
No, the 8 hours was in an animal because it was just difficult. But you know, it's not that hard. We get in the coronary sinus all the time. So that's part of a standard EP procedure, cannulating the coronary sinus. And for biventricular devices, we do venograms of the coronary sinus all the time. So, but teaching the technique is something else that requires a commitment from the operator. When we expanded the protocol to other sites, by protocol, I had to go there and proctor and teach them how to do it. One thing became very clear to me. If the operator was interested in this, they would make it happen. If the operator just wanted to say that he was part of the trial and then put 2 patients that they enrolled 2 patients when I was there and they never enroll anymore. Yeah. And that is a problem. I don't think the procedure is difficult, especially when you go from the IJ with internal mammary catheter, it just engages in the veno-martial. Sometimes it is if the veno-martial is small, but it requires a commitment from the operator. And once you have that, everything goes easy.
So what was the average procedural time for the
duration? The veno-martial adds, in the original experience, was 45 minutes of procedure time and about 8 minutes of fluoro. Right now, when I do it in clinical cases out of the studies, like the fellow is doing a map of the left atrium and simultaneously I'm getting in the veno-martial. By the time he's done, I'm ready to inject. It really doesn't. It doesn't. So it adds maybe 20 minutes, but not that much.
So you've got it pretty well streamlined now, huh? All right. Okay, well that's great. I mean, I think this, again, this was a phenomenal overview and really kind of telling us about what you've been through the last 10 or 12 years. I think the lessons you've learned, but also I think this is really a lesson in persistence, if nothing else, because I think there's many steps along the way where it's easy to just say give up and say this is the end of it.
Yeah,
and I'm done.
The prospect of having to return NIH money to me was just not acceptable. I will just, if I have to die, I'll die killing. I put up a fight, and it paid off. And the truth is that I couldn't have done it if I didn't have allies. And people are really, even administrators that I criticize came to the rescue. Even I mentioned Rob Phillips, Roberta Schwartz was helpful. I would talk to them. It's like they wanted to help me. They wanted to help me. So you have to be able to transmit that passion, because if of course they're not going to side with me if there is a fight with their own administration, right? But they wanted to help me, and you gotta find ways to let them help you, and it worked.
All right.
So, the usual time frame to complete a trial. What you tell the NIH is that in 4 years you complete enrollment, and in 1 year you complete the follow-up for a 1-year follow-up. So it depends on the trial. For this was a 1-year follow-up study, and we wanted to enroll 340 patients. So we said in 5 years we would complete. It took us 6. But the NIH can actually give you a no-cost extension, and when we continue, so it was not a big deal.
All right. Well, great. We enjoyed it very much, Miguel, and we look forward to having you back again at a future date. Thank you.
Thank you. All right. So this was the connector.